Hello everyone. In this presentation, I am going to explain the general characters of Mycoplasma. Mycoplasma are a group of the smallest free living organisms. The presentation includes the introduction, morphology and cultural characters, classification and diseases caused by the Mycoplasma. In the introduction part, Mycoplasma were first identified by Louis Pasteur, but they were isolated by Knockaert and Raux in 1898 in bovine pneumonia in the animals which were infected with the bovine pneumonia, and these organisms were named as PPLOs. PPLO means pneumonia like organisms, and later this name was replaced by Mycoplasma. Myco means the filaments, plasma means the plasticity of the shape, because these organisms, they do not possess a cell wall, that means so they do not have a fixed shape. Cell wall in the bacteria gives a rigidity and the shape, but these prokaryotes, they do not possess a cell wall, they do not have a fixed shape. Plasticity, that refers to the pleomorphism, that's why they exist in different shapes. Initially these organisms were placed in the phylum Firmicutes, but now the fourth volume of Burgess manual included these organisms in the phylum Tenericutes. These organisms, they do not possess a cell wall and they do not contain the precursors for the cell wall synthesis. Cell wall in the bacteria is generally made up of the peptidoglycan layer. So the precursors for the cell wall synthesis are also not present in these organisms. Now they are placed in the class Mollicutes. The members of the Mollicutes are called as a Mycoplasma. Moving to the morphology, Mycoplasma, they are smallest organisms. They can pass through the bacterial filters and they contain a three-layered membrane, cellular membrane, and this membrane is made up of lipoproteins. Inside the cytoplasm, it contains both DNA and RNA, and 70S ribosomes are present, and they do not possess a cell wall, and they are gram negative if they are stained, but they can be better stained by a Giamsa staining. They are the smallest organisms, so they can pass through the filters, bacterial filters, with the pore size ranges from 450 nanometers even. These organisms are present in the form of granules and in the filaments of various sizes. If they are present in the granules, they may be of cocoid, star shape or disc and ring shape. They are filamentous. The filaments are slender and they show the true branching, and multiplication in these organisms is by means of binary fission. They do not contain any spores, and flagella, even fimbriae, but these organisms, they show the motility, which is a gliding motility. The gliding motility enables these organisms to move across the solid surfaces. One such example, Mycoplasma mobile, which could move at the steady pace of 2 to 5 micrometers per second, and this movement is mediated by the cell surface proteins, and the energy is derived from the hydrolysis of ATP. Another organism like Mycoplasma genitalium, this organism has a special terminal structures that can project and appear in the form of a flask or pear shaped, and these structures, they help the organism to attach to the host surface, and the genome in these organisms is very small, and that would range from 0.7 to 1.7 MB and contain very less number of genes, that is around 1000 genes. Now moving on to the cultural and biochemical characters, The mycoplasmas can be cultivated on fluid and solid medium. They are generally facultative anaerobes. They can survive in the absence of oxygen, but a few microorganisms are obligate anaerobes. That means they can live only in the absence of oxygen. On agar medium, they produce the colonies. After 2 to 6 days, the generation time is slow and the colonies will be in the form of fried egg appearance. That means here the central region is opaque and it is a granular in appearance, and it extends into the depth of the medium, and this which is surrounded by a translucent flat peripheral zone. So the colonies are generally seen with a hand lens and observed by means of a staining, that's by a Dion's method. This is suggested by Stevens and Fox in 1979. In this method, the block of agar containing the colony is cut and it is placed on a slide and it is covered with cover slip on which the alcoholic solution of methylene blue and azer were added and dried. The colonies here cannot be picked up with the loops, but agar block containing the colonies will be rubbed on the surface of the fresh plates for subculturing. And the media are enriched with 20% of the horse serum and yeast extract. The serum provides the cholesterol and the related sterols which are incorporated into the plasma membrane to facilitate the osmotic stability. And the temperature is about the 22 to 41 degrees centigrade. The optimum is about 35 to 37 degrees centigrade. In the medium, penicillin and thallium acetate can be added as a selective agents. And the organisms are chemoorganotrophs. That means they show the fermentative metabolism and they produce energy in the form of ATP by EMP pathway, that is Embeden-Meyerhof-Parnas pathway, which is a glycolytic pathway, and the lactic acid fermentation. These organisms, they utilize glucose or arginine as a major source of energy, and they lack the ability to synthesize purines and pyrimidines. These organisms, they contain the genome which is very small in size. It ranges from 0.7 to 1.7 MB. These organisms, they possess a minimal genome size for the free living existence. The limited number of genes in these organisms shows that they are enabled to synthesize a number of macromolecules. For example, Mycoplasma genitalium, it depends heavily on the biosynthetic capacity of the host. It does not contain the genes that synthesizes and enzymes which are needed for the amino acids, purines, pyrimidines and fatty acids. Small size of genome made these organisms to be a model organisms for the development of a synthetic bacterium. Synthetic bacterium means here the bacterium can be constructed with a genome that is synthesized chemically in the laboratory. Moving on to the cell structure, they are prokaryotic cell structure, that means they contain both DNA and RNA and 70S ribosomes with ribosomes with 40 nanometers in diameter. DNA is in the form of double stranded helical nature. The G+C content is very low and it is distinguished from the bacteria by means of having the low G+C content that ranges from 23 to 46%. The generation time is generally slow, that requires 2 to 6 days to produce a colony, and the mode of multiplication in this organism is a budding or the binary fission, and as the cell wall is not present and they are resistant to the antibiotics like penicillin, cephalosporin and vancomycin. These antibiotics are the cell wall inhibitors. So these organisms are resistant to the cell wall inhibitor drugs. Now moving on to the classification, these organisms are placed in the phylum Pterygoids, order Mycoplasmatales, and class Molecules. It includes 4 families. They are Mycoplasmataceae, family Echolplasmataceae, another third one is Spiroplasmataceae, Anaeroplasmataceae. Mycoplasmataceae, They are the parasitic in nature. The family includes the organisms with parasitic in nature, and they require the cholesterol or the other sterols for their growth, and includes two genera. One is a Mycoplasma, another one is a Ureaplasma. Mycoplasma utilizes the glucose or arginine, but they do not hydrolyze the urea. Ureaplasma is one genera that could utilize the urea, that means utilize it for the growth. Another family, the second family is Echolplasmataceae. Echol, that means they do not require cholesterols or the sterols, and they are saprophytic in nature. And Spiroplasmataceae, the shape is of a spiral in shape, and they require the sterols for their growth. Usually they are the parasites of arthropods and plants. Anaeroplasmataceae is the another family, so which contains anaerobic organisms. They are the strict anaerobic organisms that found in the rumen of the animals. Rumen is a specialized structure where the anaerobic environment is present for the digestion process in the animals of cattle and sheep. They can be isolated from animals, plants, and the soil, and even in the compost piles, and they can cause diseases in plants, animals, and human beings. In animals, Mycoplasma colonize the mucous membranes and joints, and they are often associated with the respiratory and urinogenital infections. Mycoplasmas, they cause several diseases to livestock even. Examples are Mycoplasma mycoids, which cause a chronic respiratory disease in chickens, and Mycoplasma gallisepticum, it causes a pneumonia in swine. And in plant diseases that are caused by Mycoplasma, examples of little leaf disease of brinjal, bunchy top of papaya, which is broom of legumes, yellow dwarf of tobacco, sandal spike disease, stripe disease of sugarcane, which is broom of potato, etc. In the human beings, Mycoplasma pneumonia causes the disease of primary atypical pneumonia. This is called a walking pneumonia. And Mycoplasma hominis and Ureaplasma ureolyticum, and they cause genital infections. Even in Ureaplasma ureolyticum is associated with the premature newborns, and the meningitis, inflammation of meninges in the neonates, and even pneumonia. Mycoplasma oral and Mycoplasma salivarium are found responsible for the respiratory tract infections. Mycoplasma have been also found in the cases of arthritis and in the inflammation of the middle ear. So Mycoplasma, they could cause the diseases to plants, animals, and human beings. Even Spiroplasma are associated with the plant diseases, which could be transmitted by means of arthropod vectors. Summarizing the content, these are the organisms which do not possess a cell wall, and the smallest living organisms, they could pass through the filters, bacterial filters. They require the cholesterol and sterols for their growth. They contain the three-layered plasma membrane, which are made up of lipoproteins. They are resistant to penicillin and the cell wall inhibitor drugs. They could cause diseases to plants, animals, and human beings. They can be cultured on the solid and liquid medium, which can be enriched with the serum and the yeast extract, etc. So, thank you for watching this video.